So question.
Yeah, Brad Ozenberg at NIH. In the great projects at England and Australia, it wasn't clear to me what your strategies are for returning secondary information, the non-diagnostic. How far are you going to go in the return of secondary genetic information?
Yeah, I'll start with what we've done from in the Melbourne Genomics project, is that we very much did a targeted analysis of the genomic sequence that was in regard to the specific clinical presentation. So we used it as a clinical test, and the patients then had the option that if they wish to, for example, look at the ACMG recommended secondary analysis, that we do that as a separate clinical face-to-face and a separate counselling. So we've done it within a clinical setting. We're using it in a more targeted way, but the patients are given the option for that broader analysis.
And will you go to polygenic risk or carrier status or other things?
Well, it's all possible on interrogating the data in that way, but for this particular, for the data that we presented today, it is it's a targeted analysis within clinical care. Because that, I think you do need to have your informed consent in a separate way for looking at different, the non-clinical indications.
So just very quickly, from an England perspective, in the 100,000 Genomes project, there's, I think, 12 conditions that are included for additional finding feedback. If patients consent to that, we've not started to feed that back, but that's the starting point for the new NHS, National Health Service system. But we are having a set of ongoing discussions with our experts and clinicians about what the future should look like and whether we should go as far as the American guidelines outlined, but recognising all the inherent issues and additional resources that this may place on a health system. So anything we do would have to be on the basis of doing a health economic assessment alongside it.
Folks should come to microphones while people are considering that. David, I want to ask you a question, going back to the first talk in the session. You know, I think you did a very nice job trying to simplify the process by which these things are going to be built up, even though I recognize under the hood it's incredibly complicated. In general, the folks that you're working with, are you sort of modeling this or using as a framework other examples where this has been done in other sectors, not even necessarily biology, where lessons were learned, or are you really doing these steps almost de novo and inventing it as you go without any pre-existing experience to rely on?
That's a good question. I haven't thought about it that way. I think there are probably two areas where we're very much tapping into experience, but not so much saying, oh, let's emulate exactly that. One area is just around the process of building a platform for a community, and things like Ewan always references W3C, and there's a mantra we used to use a lot as we were working together of rough consensus and running code, and just the process of taking a new area that we knew needed to have lots of different things snapped together that wasn't really clear, right? So there's a process bit. The other bit is just there's a lot of the arbitrary decisions around how you package up technology to snap together, where there's lots of that where there's no reason to invent it, right? It's like, you know, if the way to specify an API programmatically, if you're going to say exactly how my code is going to call Kathryn's code to talk, we don't have to invent a way to specify an API. There are 2 or 3 choices. We can either use Proto 3, we can use Proto 3 wrapped with Swagger, but you just pick one of the standard ones and use it. You know, how should we collaborate on code? Oh, well, we'll just use GitHub repos because that, right? So there's a whole set of the mechanics that we can just take.
So you pull off the shelf when necessary. So it's a little bit of both is what I'm hearing. Okay, that's helpful. Question, Terry?
Terry Manolio from NHGRI. So it was fascinating to hear how closely integrated the clinical and research enterprises in England are, and I was wondering if the other speakers could comment a bit, maybe Kathryn, and heaven knows in the U.S. integrating anything is a challenge, but thoughts you might have as to how we could increase that integration within those healthcare systems as we're trying to do this research.
In terms of the Australian system, we, as I mentioned, we've set up a national functional genomics network that one will serve the ability to look at variants of unknown significance in terms of working out pathogenicity, but also by screening known genes, we're also producing enriched cohorts for gene discovery. So we're now building within that system the network where we can start to do pathway analysis or use a smorgasbord of different approaches that will actually accelerate the ability to do that sort of functional genomics disease mechanism leading into drug discovery. So that is the layer that's really quite closely linked now, I think, with the clinical networks.
And I'll also add, just as we think about platforms like Matchmaker Exchange or ClinVar, these platforms are really being used today on the clinical and research divide, in the sense that physicians are ordering exomes on patients. A lab will report back a candidate gene in a clinical report, and the physician will immediately enter that into a Matchmaker Exchange node, leading often to a match to find another patient. And that's, in essence, doing research and clinical care all in one shot. And I would argue that's what we've been doing for eternity in variant interpretation. When we find a variant, we don't know what it means. We search all the databases. Sometimes we find that someone else is deposited somewhere. We reach out, we contact that group, we find out what they know, and we incorporate that into the evolving knowledge on that variant. To me, that's no different than research. You're doing research in real time with clinical care.
Okay, in the interest of time, I think I'm going to wrap up this session. We have another round of applause for our 4 speakers.